Let's talk about this. Okay, I'm going to try to change. I'm sorry, online students, I'm sorry about the reflection, but I hope you'll be able to see, after I put the screen down, you'll be able to see things that I didn't write. I mean, that you couldn't see when I wrote it. So, what goes in box number 1? Anybody? You know what we've been talking about. Zoe?
The action potential arise in the presynaptic axon terminal.
Exactly. Action potential propagation. So, step 1 is the action potential arrives by propagation at the presynaptic terminal. Okay? What happens next? What happens in box number 2? Or 2 and 3, they're kind of lumped together, so let's not be careful about that. Henry?
So does that trigger the release of the potassium, or not potassium, the calcium gated channel?
Say that again? You're close.
Does that so like basically the action potential from the previous one opens the calcium channel?
Exactly. So these are voltage-gated calcium channels. They're similar to voltage-gated sodium channels, but they're calcium selective, highly selective for calcium ions, and they're slow compared to voltage-gated sodium channels. But they're there. They're highly expressed in the synaptic terminal. So we open voltage-gated calcium channels. I'm just going to use that abbreviation. So step 2 is the depolarization opens, the depolarization of the action potential caused by sodium channels opens voltage-gated calcium channels. Okay, so then what's number 3? What happens when voltage-gated calcium channels are open? It's illustrated by these arrows here. What's next? Influx of calcium. Calcium influx. So the next step is calcium ion influx through voltage-gated calcium channels. So what happens to the cytosolic calcium concentration? Goes up. So increase in the concentration of calcium on the inside. What does that calcium bind to? I'm getting to step 4. What happens here? There's a name of a protein that you learned about in cell phys. I'm going to reinforce that knowledge. What is the calcium bind to? If this were a muscle, calcium would bind to troponin. But this is not a muscle, this is a nerve. So that is a calcium binding protein, but that's the calcium binding protein for muscles, which we haven't talked about. But we will. It's a good answer, but it's the wrong answer. What does calcium bind to here in this not muscle synaptic terminal? It's got a name. Synaptotagmin. Calcium binds to synaptotagmin, which is one of the B snares. So you may remember the snare complex. We're going to show that in a minute. Synaptotagmin is one of the V snares, the snares associated with vesicles. And in this case, it's got a name, synaptotagmin. Okay, let's see. So that was calcium binds, synaptotagmin. And we're going to have the snare business. It's going to happen. What is illustrated in box 5 down here? What is happening here? Well, actually, we skipped a step. So calcium binds to synaptotagmin, and we have the V snare thing going on. And what else is going to happen here in blue before we get to what happens down here in black? What is this going to cause? It's shown right here. I'm sorry, I can't hear you.
Fusion of vesicles.
Fusion of vesicles. Fusion of synaptic vesicles and release of neurotransmitters into the cleft, synaptic cleft, the space between the presynaptic neuron and the postsynaptic neuron. Then what happens? Now if I'm tracking. Then what happens here? So snare, binds, synaptotagmin, snare, fusion, vesicle release. Neurotransmitter release. What happens down here? I know you know the answer. You're just thinking. Overcome your inhibition. Give me an answer. Okay, I'll start with the first letter of the first word. Okay, you've got neurotransmitter What does the neurotransmitter do when it reaches these things over here? Henry?
It binds to the
Binds.
receptors.
Neurotransmitter binds to what? What do we call the things that it binds to? Receptors. And then the receptors activate. And now it depends on what the receptor is. If the receptor is a GABA receptor, the opening of a chloride channel happens and we might get a hyperpolarization that you learned about in the problem set. If the receptor is a GPCR, it's going to go through that whole series of events that you learned about for GS and GQ and change the excitability of the next cell. If the receptor is a acetylcholine receptor that you learned about from the problem set, sodium influx and depolarization. Anyway, the point is here, receptor are activated. And the final step here that is sort of obscure is we're going to have to talk about the reuptake of neurotransmitter. The ending. Remember, everything that begins has to have an end. And so we're going to reuptake the neurotransmitter. We're going to talk about that in a minute. So your handout should look something like this. Let's see if we got it right. Action potentials, calcium channels, calcium influx, synaptotagmin, D snares, T snares. We'll talk about conformational change in a minute. Fusion, we got the fusion idea. Release of neurotransmitter. Oh, I forgot to mention that one of my favorite processes, the neurotransmitter diffuse. Right? The neurotransmitters diffuse. They aren't sucked into the postsynaptic cell. They diffuse and bind to the receptors. I didn't cover the fact that, well, we didn't get to, I guess I ran out of space before we got to binding to postsynaptic receptors and activating them. Questions about this process?
Is the terminology calcium-induced calcium release specific to cardiac myocytes, or can it be used in this?
Calcium-induced calcium release is a special feature of cardiac myocytes. It happens in neurons, but not here very much. There is some in the presynaptic terminal. Calcium influx through voltage-gated calcium channels, triggering calcium release from the ER in the presynaptic terminal. That happens some places, specialized synapses. So it happens, but it's not as important as it is in cardiac myocytes. That was a high-level question because of someone who took AMP2 before they took AMP1. Other questions? Okay, so let's talk about the snare business just for a moment. This is reinforcing some information from cell phys. Here is a cartoon of the presynaptic membrane. This membrane here is this membrane here. This membrane is the synaptic vesicle. So this membrane here is this membrane here. The synaptic vesicle, well, maybe it's one of these that is close. This membrane and this membrane are what are cartoon blown up here in this cartoon. Here is synaptotagmin, which is part of the V snare, and this is the snare complex. So this is the V snares are the proteins that are needed for exocytosis that are associated with the vesicle, and synaptotagmin is the one that's specialized in neurotransmitter exocytosis in neurons, and it has a calcium binding domain. The V snares and the T snares, T stands for target, for the target membrane into which this vesicle is going to fuse, form this coiled coil and dock this vesicle very, very close, like nanometers from the membrane that it's going to fuse to. And so in cartoon version, we've got V snares, including synaptotagmin, T snares that have names that I'm not going to ask you to learn, but they have names. And in fact, this is not all of them. And we have voltage-gated calcium channels that are localized pretty nearby. When the vesicle interacts with through this coil interaction, the vesicle is docked. It's very, very close. It's ready to fuse. So what that is, that's cartooned here by these vesicles that are docked at the site, ready to fuse. And they're in sort of an equilibrium population with vesicles that are sort of in reserve, ready to replace these once these fuse in. Anyway, this vesicle is docked, ready to fuse. Voltage-gated calcium channels, calcium influx, calcium binding to synaptotagmin, conformational change of the V snares and T snares through protein-protein interactions, a cinching and a fusing of the vesicle with the cell membrane, and diffusion, right? This is not being squeezed like a balloon or anything. There's no force here. This is just diffusion of the vesicle. Diffusion of the neurotransmitter from the vesicle into the synaptic cleft. And then there's going to be recycling of this. This is then, like everything else, what turns on has to turn off, and endocytosis of this vesicle is going to happen, but it's not shown here. So there's an interesting factoid. This will not be on any test, but an interesting factoid is the toxins in botulinum. So if you get botulism, which you may, if you know, it's a neurotoxin. It's a paralytic neurotoxin. Botulism prevents neurotransmitter release by cutting the snares. So different toxins have different cutting sites on this process and prevent neurotransmitter release. So poisoning by botulinum toxin causes death by respiratory paralysis. Injecting of small amounts of botulinum toxin into the muscles of the eye prevent the person from frowning and make them look young and happy again. That's Botox. Botox prevents neurotransmitter release in the muscles where it is injected. Obviously, not a lethal dose. A very, very small, localized dose of botulinum toxin is Botox. So now you know. So let's do another. So we're going to take a break, and you're going to do another right exercise. I want you to remember what we talked about last week about the processes, not for endocytosis of the membrane. That self is, you remember endocytosis, or you could. I want you to remember from last week the transport mechanisms necessary to reuptake and repackage the neurotransmitter into vesicles. Trying to get you to recall information from just a week ago. So we're going to take a break, work on this problem. Maybe in 10 minutes we'll get back and reveal it.